I did expect you to.
So,
No problem, please.
right, let me check which ones. Okay, so this is not going to be a presentation because I haven't prepared one, but I have slides that explain the difference between alcohol 1, alcohol 2, and alcohol 3 principles and why. So if I share, can you see? Okay, perfect. Right. And so these two things were really critical. Once it was the problem was defined that we wanted to full proteins. So this is work from Cyrus Chotia and Arthur Lesk, who are here in Cambridge, and what they showed. This is from 1986. So very few structures. But what they showed was that structures are more from 1.2 angstrom. So that's what this is saying. So this was the first kind of principle. And then in mid-90s, so this is 1994, another kind of hypothesis that given that sequences fold in 3D structure, what must happen is if I change one residue, then if there is another residue which is in contact with it in 3 dimensions, then that needs to kind of accommodate that change. And so this is the co-evolution principle that, if you have to keep the structure, then there needs to be compensatory mutations happening when something changes in sequence. So these are the kind of two principles that are used, and then we have AlphaFold. But what I want to explain a little bit is these the metric that it provides, both the PLDT and the PAE. So why we need this is because when we have a prediction, we want to know what is the confidence of the AI method itself in a given prediction. And so this is what PLDT is about. So the LDDT stands for the local distance difference test. What it means is it's looking at local packing. Is the residue happy in its environment? And that's what PLDT is about. And so the higher the PLDT, the happier the residue in its local environment. And so this is what this is showing. And the lower the PLDT, it can be interpreted in two different ways. One interpretation could be that the method was not able to predict the structure. The other interpretation is that it is a disordered region, so it doesn't have a structure. And it's been shown that when you analyze these structures and the low PLDT regions, it matches really well with our predictions for disordered regions that were there before this. And so, just one clarification that AlphaFold is not designed to predict disorder. It is designed to predict order, but it is so good at predicting order that when it doesn't predict order, you can kind of think that region might be disordered. So, but there will be cases where AlphaFold fails to predict order and structure. So So just be careful when you use this. Now what we can do with this is to obviously identify the domains and see where they are and then look at the function, etc., etc. So this is for individual residues, and proteins are made of many different domains sometimes, and you also then want to understand the metric confidence in the relative position of the residues, and this is what the PAE. Oh, okay. These are the pitfalls. Yeah, this is what I just said. But PAE gives you that metric that allows you to check whether the method has high confidence in the prediction of relative position of two residues. So this is just two examples where in the same domain, so this is the same example we used before. If these two residues you see here, it's green, and what that means is the error in the relative position is low, right? So if it is green, that means the relative position is also predicted with very high confidence, and if it goes towards white, the relative position is not predicted with high confidence. And you can see why, because these two domains could be anywhere in space, and this is just one confirmation that you might get for this particular protein. And so this is what the PAE tells you. So why AlphaFold 2 was so much better than AlphaFold 1? So AlphaFold 1 was based on the idea that you kind of get information about distances between residues using the coevolution principle, and then use that to predict the structure. So it's exactly like in the NMR experiment, you get NOEs, which is giving you distances, and then you predict the structure from it. But obviously, there are issues with that. You have to optimize all the chain to get the right structure for all these distances, and you can never ever satisfy all the distances. And so that's one kind of issue with how AlphaFold 1 worked. But AlphaFold 2 was designed to kind of more reflect the physical principles and geometry of proteins. So it does not, first of all, it doesn't say that residues are connected, it allows them to move freely. And so there are no constraints in terms of the movement of one residue affecting the other residue. It allows the relationship between close residues to evolve as the method works, right? So the backbone connectivity obviously evolves, and so that's one thing that you could get out of it, and you can then be confident that actually it has worked because it did give kind of right backbone for a given protein. And what it does then is, because it is free to move, it can optimize the constraints between residues that are close to each other and residues that are far away from each other. And that's how the kind of structure evolves better. And then you can optimize it for model length angles, etc., etc. But that's one of the reasons why AlphaFold 2 kind of works, because it integrates this kind of geometric insights of how the proteins work in the network. And so what it does basically is predicts global structure and local structure in a way that optimizes both. And that's a really important part of why AlphaFold 2 is so good at predicting structures. And the side chains typically are predicted well if the backbone is predicted well. But you can also think about, if the PADDT is high, that means the residue is happy in its environment. And so obviously it will pack well, and to pack well, the side chain has to be optimized as well in a way. So that's what is happening here. The other thing, which is true for experimental structures as well, in NMR, you have to get the right LOEs, right? So you have to get the right constraints to get the right structure. And so some of these very few constraints actually drive the whole global structure as well. And so you can kind of say locally AlphaFold understands the physical and geometric constraint because the loss function is about PADT as well. So in a way you are optimizing these things. Right? And it doesn't have any physical principles. So no free energies, no kind of optimization, anything like that. So keep that in mind. Okay, let's go to just AlphaFold 3. What is the difference? This bit is more or less similar. What is different is how the structure is predicted, and this is diffusion model. And diffusion model has a property that it tries. So diffusion model is basic, or diffusion. The way it works is you have a picture, let's say, with lots of noise, and it denoises it to get the original picture. But it has the tendency to try and get that structure. And it tries really hard to get it, right? And this is what is called hallucination, because it's trying its best to get a structure, in this case, from the noisy data that is provided to it. And so what that means is if you take a protein complex and predict it with AlphaFold multimer, you might see something like this. Whereas if you predict it with AlphaFold 3, you will see something like this, and it's trying its hardest to really get structure, right? And the reason this happens is because in the training set, which is PDP, we don't have disorder. It hasn't seen any disorder. And so obviously it's trying to mimic its training as much as it can, learn from its training as much as it can. And so it tries to get as much structure, and this is why you see all these helices appearing with very low PADTs and things like that. So this is what is hallucination in this case. So how do you get rid of that? What you want is, in the training set, you want to have disorder, so that the network also learns about disorder. And obviously in PDP we don't have it. So where can we get it? We have 214 million structures from AlphaFold itself. And so you can take all the AlphaFold database and you can learn from that how disorder works. And so that's what happened, that's what they did. And AlphaFold 3 was trained also using the AlphaFold database. And then suddenly you start seeing really good structures from AlphaFold 3 as well. And it's not hallucinating as much, it's still hallucinating, but not as much as it would if it was not trained using the AlphaFold database. So in AI speaks, this is called distillation. So this is learning from what you already know, basically. And that's how AlphaFold 3 works. And you may have seen these slides already. I don't want to go into that. But I want to say, let me check. Yeah, maybe this one. Okay. So it has the same kind of confidence metrics as PADT and PAE, and they have very similar meaning. But it also obviously has the interface related score, the IPTM, which is the interface TM score. And so one of the things to remember about AlphaFold 3 is that it's very sensitive to its context, right? And the reason being, it's trying to get the whole structure. So the more it knows about the structure, the better structure it is going to get. And so if we see, for example, this transmembrane domain, AlphaFold database has this structure, AlphaFold 3 has this structure. But if you kind of provide it with the context of the membrane, which is this adding the 30 oleic acid, then suddenly it understands that this is a transmembrane. And then it puts it in that way. And so you can also see the PADT is improved for the whole structure, right? So the more context it can have, the better structures it will produce. And this is different about the whole too. And so, for example, another example where there was a metal binding site, and so suddenly when you add metals, which are here, you get a better structure because it now knows the more context. And so this is why alcohol 3 also changes PADVTs depending on the inputs that you give, because the more complete the context, the better prediction it is going to give you. So this is another example that is being provided, and you can see here it's all kind of low PADVT, the PAE doesn't look that good, but once you start providing the metals and other things, it improves a lot, and you provide the membrane, it improves in the bone, etc. So you need to kind of think about alcohol 3 very carefully, because what you put in is really important and affects what you get out of it as well. This is an example of what not to do. And this is a paper which was based on predicting alcohol 3 structure, and they talked about phosphorylation, etc. The first thing you see here is they have colored it in completely different colors, and the PADT and things are not shown at all. And if you go and see what it is, it looks rubbish, completely nonsense. And so this whole paper is based on this prediction, and you can't really take anything from this. It's complete nonsense. So which one should you use? Obviously, you have to look at the license terms. So alcohol 3 is only for non-commercial use, and read the license carefully. You cannot use it for commercial activities, train other models, etc., etc. I would use alcohol 2 if I'm just worried about getting proteins, and I will only go to alcohol 3 if I have nucleic acid or metals or ligands, etc., etc. Or antibodies. Alcohol 3 is better at predicting antibody complexes as well. But if it was just about proteins and even protein complexes, I would try alcohol 2 and alcohol 3 also, and not just by default go to alcohol 3 because it's higher, and you think it might be better. So I think I'll stop there.
Thank you so much, Samir. I believe you have just delivered a great summary, and I just want to give the floor to the participants if they have any questions. Anyone? Question?
Can you hear me?
Yeah.
So you mentioned that AlphaFold 3 was trained on the disordered regions from AlphaFold 2 results.
Yeah.
So how is it that you train to learn disorder? Because the nature of disorder is that it doesn't have a stable structure. So how do you learn something that is not stable? And
Well,
then how do you what's your reference to make that comparison to then validate the AlphaFold 3 output that you get?
Yeah, okay. That's a really good question. And as I said, the disorder part is really coming out as a consequence of predicting order. So that's the first thing. So with AlphaFold 2, the database, what the module, when you train it with those structures, what it is really learning is where are these disordered regions, and for the diffusion model, it it's also learning that having disorder is fine, that you don't have to look for order everywhere, if you see what I mean, right? So that's what is happening. So it's training it to understand that proteins have disordered regions and proteins have ordered regions, and that's about it. So it's not kind of predicting the disorder. It is still predicting order, but now the order it predicts is slightly more confident, and you can kind of look at it and use it better because it also understands that there is disordered regions. Does that make sense? Yeah? Okay.
Thank you. We're delighted for having you among us. And I actually am curious about the future direction of the future. What paradigm shift may AlphaFold, the next AlphaFold may bring?
Okay.
Yeah. Thank you.
Right. So what are the next questions? Basically is what you're asking. And I think we still don't know a lot, right? We don't know how the protein dynamics works. So different conformational states, we still can't really predict the function just from the protein. So these are the kind of things people are learning. AlphaFold actually, even predicting complexes is not a solved problem. And if you look at how the difference between what we get with AlphaFold 2, where we are single chain predictions and they really are very good when you compare them with experiments, the complexes are much more difficult because there is dynamics involved when you form a complex, but also the interfaces, and we don't have enough data on both these things yet. And why I say that is because when AlphaFold Multimer was retrained with using more structures from the PDP, it improved the prediction of complexes. And what that tells us is the diversity of data that you have in the PDP is not sufficient yet for it to learn all the possible ways it can make interfaces. And so predicting complexes is still unsolved problem, but on top of it, it is complicated because of dynamics, and we are still getting a snapshot of single things, right? We need really to understand protein function. We need to understand not just what the protein is doing, but what are the different dynamic conformational states, etc., etc. So those problems are still problems that people are working on quite a lot. Obviously, ligands is another. Nucleic acid, we don't have sufficient data, and so nucleic acid prediction and nucleic acid complex prediction is still I would say, at early stages. Antibodies is another thing because you can't use the full evolution principle there. So there are quite a few things that still need a lot of work.
So can I ask a complementary question?
Yeah.
That is actually, so we are actually heading towards unifying the structural prediction with the dynamics and also the function in a single framework. That's the direction we are heading towards, right?
Yes, that would be fantastic if we can do that.
So if we envision that, how long can it take? Just curious, actually.
That will depend on a lot of things. That will depend on how much data we have, but also probably some of the AI methods need to improve as well. And so I don't know. I never thought I will see AlphaFold in my lifetime. So I have no clue how much time it will take. Maybe tomorrow we might hear something. Who knows? But I think you also need to think about this. This is still, we are talking about single proteins or complexes, etc. The real thing to understand is how in their context, right? So in the cell where these things are, because that makes a difference. All the different isoforms, variants, that makes a difference. So biology is so complex that there are many layers to it, and we're still just scratching the surface, and there is a lot. Yeah, I can see it.
Okay, so if I'm working with a protein sequence and I want to predict the structure, so I get a structure from AlphaFold 3. Now I want to know if the structure is really reliable or not. Previously, when we worked with Swiss model or IGAS or Python, we used to get the sequence and we used to validate it with some other tools like Growth or Verified 3D or Project. In case of AlphaFold, the 2 or 3 predicted structures, do we need to validate the predicted structures with other external tools?
Well, so what you are talking about is just the validation of the geometry of the structure. That's not validating whether the structure itself is correct or not. What it is validating is the structure looking like a protein, right? So project or all the things that you mentioned are just telling you that this thing looks like any other protein. It doesn't tell you whether it is the right structure for that protein or not. What we still need, and maybe I should show the next slide. What we still need is validation of the predicted model. And what that means is, obviously, we can do it in different ways. We cannot solve every structure. But what we can do is, if we have other types of data, then we can see if the predicted model can explain that data. So, for example, if you have a mutation data, or you know that certain residue, when it is changed, it disrupts the function or structure or something like that, you can look at the prediction and ask that question: can I actually derive the same conclusion if I look at this structure? And so, what we are trying to do actually is to look at huge amount of data in literature to see, can we extract that data, can we understand the residue level annotations, and can we then use that to validate the protein structure. And this I call functional validation. This is not validation of the geometry of the protein. It is validating whether the actual structure is right or not. So you should never just depend on, because project gives you good answer, doesn't mean the structure is right. You have to then design experiment to say, what can I do with this structure, and what happens if this structure is correct? What is it telling me that I can validate experimentally?
Okay, thank you.
What you have to remember is all these methods now are so good at getting the right Ramachandran angles and things like that, that running project or ball probability or things like that is not necessarily going to tell you whether the structure is good or right or not. It's only going to tell you whether it's protein or not.
Okay, so I have a question. We are seeing that models are getting smaller day by day, and especially in case of vision models, we can see that we can run billions of models having billions of parameters in mobile devices. So does the community dream about predicting proteins or other complexes using just mobile devices in maybe 20 years or 30 years?
Well, who knows? Yeah, probably. I mean, this is the thing. Things are changing so fast that even predicting what's going to happen is difficult. But I think the big takeaway message is all these things are still tools, right, that we need to use to do something with them. These are things that might help us do our research better, or it might make it more efficient, and that's the direction. So you see all these agentic things and lab in the loop concepts and things like that, where the AI agents are driving experiments, etc. So what you have to remember is, at least right now, all these things are using the knowledge that we have, and what it can do better maybe than us at the moment is make the connections that we may not make, because we can't just read everything and understand it. But what it doesn't do is to come up with completely new thing. It makes really good connections, and I think that's valuable, and we will learn a lot from that. But it's still not doing things which are completely new, out of the box thinking that humans do, at least right now.
Thank you.
So I think no question left. So we can conclude, but before that I would like to have a picture. You will be there obviously, but maybe we can come over here and maybe you can be seated in the backward. So I just request everyone to come here and take a photo. We didn't have any photos so far. So it is also important. So please come here.
রেডি এদিকে সবাই
Hi,
আপনি একটা ছবি স্যার আপনি নেবেন